Hello everyone and welcome to the class. The topic is enzyme. But before coming to the topic or discussing much more about it, first of all, we'll learn what is an enzyme and why we require it. Okay. So first of all, we are a biological system, and for uh, running this machine, what we need is energy, and this energy is being derived from the biochemical reaction, and this biochemical reaction may involve the breaking down of or maybe making of certain biomolecules. So you know what are the biomolecules. Okay. And for this biochemical reaction to be fastened, we require biocatalyst, or we require these reactions to be catalyzed, and the proteins that catalyzes this reaction are enzymes. Okay, so basically we need to know enzymes so that we can have an efficient system. This is the need of enzyme. And what is a catalyst exactly then? So catalyst is, it's a chemical, and it is responsible for increasing the rate of reaction or decreasing the activation energy that is involved, but it does not get consumed in the process. And similar is the nature of enzymes, and hence they are known as the biocatalyst. Just a moment. Okay, let's continue. And sorry for the break. I have something to sort. Okay, chalo, move on. So this is what biochemical is, and that's why we are calling enzyme as biocatalyst. Sorry, not biochemical. So then this is we can say it is a way the nature had evolved us in such a manner that it can um, we can on our own uh, do chemical reaction within us, we do in lab. Maybe it's sounding funny, but I think that's quite similar situation. Ki, a lab, we are simply mixing up certain chemicals, and then maybe if it required heat, we are heating it up and adding certain other chemicals, metals, so that we can fasten its rate and get our required result. And similar is the case key in our body as well. At normal temperature and pH, there is this biocatalyst functioning with its optimal capacity to give us the required product. Okay. And a catalyst, uh, maybe your inorganic catalyst or enzymatic catalyst. So the difference is ki inorganic catalyst, they work at any temperature and pressure. For example, the potassium permanganate. But when it comes to enzyme catalyst, they get damaged at high temperature, and hence they are known as thermo liable. That means they are sensitive to temperature, or there are certain character or features that uh, can hamper this enzyme. And what are the changes? The changes may be uh, in physical, that is change of shape, or the state of matter, or maybe chemical, the breaking of bonds or making of bonds. This is what the enzyme basically do. So first, who discovered the enzyme? So enzyme is being discovered, or this term was coined by Kuhne, and the proteinaceous nature was discovered by Sumner. And the first enzyme which is isolated is zymage by Bunchner. This four name are quite important. Some can ask you, okay? And the branch that deals with the study is enzymology. And as you know, all enzymes are protein, but reverse is not true always. And 70 to 75% of enzymes are there in mitochondria. Okay, that's it. Next, the types. So when it comes to types, so there can be non-protein and protein. So protein, they are made up of only proteins or protein along with some other things. And then comes your non-protein, and these are not proteinaceous in nature. Example is your RNAs and ribozymes. Fine. Then this protein enzyme again of two type. First is your pure protein, then the next one is your conjugate protein. Sorry, conjugate enzyme and pure protein enzyme. Pure protein are only protein, and conjugate protein along with some other things. For example, that may be a coenzyme. So basically, what it contains, one protein part which is known as the apoenzyme and one non-protein part which is known as coenzyme. Together they are forming the holoenzyme. Okay, and depending upon their action, if functioning within, that is endoenzyme, and if outside, then exoenzyme. Then structure. So structure, it's having two structure, and they are basically globular proteins, and uh, the enzymes are required in very small amount. So basically this cofactor is again of three types. They may be um, very simple inorganic compound, or maybe complex organic molecules, and depending upon this, they are again divided into three types. So first is your prosthetic group, coenzyme and metal activator. And this prosthetic group and coenzyme are your organic compounds. So prosthetic, it's derived from a Greek word which means addition. And this prosthetic group are tightly bound to the apoenzyme, and they are on a permanent basis. Okay. For example, as I mentioned here, that is the porphyrin part of heme protein in catalase. Then comes your coenzyme. So they are loosely attached and attached when at the time of reaction. And the example of coenzyme is vitamins. That's why the moral, or the important thing is we need to consume vitamins for the proper functioning of our body. Next comes your metal activator. So metal activator, there are several metal activators, like these are inorganic substances. For example, your manganese, magnesium, copper, zinc, molybdenum, iron. So these are some metal activator, and if we like see whether they are doing or not. So molybdenum, which is a metal activator, uh, it is required for the activity of nitrate reductase. Okay, and this is how the structure is. And apart from the composition, these are the parts, and where these parts located, there are certain sites that are present in an enzyme, and these sites are your active site and allosteric site. So active site, it is the part where the substrate binds, and allosteric site, it is a receptor site, and it is a site which is other than the um, active site, and when it binds, it brings change in the uh, enzyme, and hence it acts as a regulator or modulator, and they can be act as activator or your inhibitors. And the site where they are getting generated are the ribosomes. And then comes the property of your enzymes. So the first thing to know that they are colloids in nature, and the next one that they are very much specific, that is they are stereo specific, and they are required in very few amount, or very low amount is sufficient. And these enzymes only change the rate of reaction and not the direction of equilibrium, and as well they are not getting consumed in the whole process. Okay, and the this enzyme, they are you know uh, they are there to lower the activation energy without any alteration. And this is like all the things, and uh, they are having a particular state of enzymatic and uh, physical properties depending upon their sequence as well, and also depending on key what are their characteristic. They may have uh, different optimal temperature and pH as well. And three factors that are affecting it: first is temperature, pH, and substrate concentration. So if we keep on increasing the temperature, the rate of reaction will increase up to certain level, and after that they will decline. And beyond the temperature, that means at very low temperature or at very high temperature, the enzyme action will be very low. And similar with pH, uh, at a particular pH they will perform optimally, and other than that there will be variations. And substrate concentration, when it's reaching the saturation point with the substrate, then the rate of reaction will be steady. And the curve, as you can see, it's having a straight line, this green one. So this is how the factors affecting our enzyme. Okay. And next comes to uh, the Michaelis constant. So it was proposed by Michaelis and Menton, and both Michaeli and Menton, what they did, uh, they provided a constant, uh, so that the substrate concentration and its velocity and the rate of reaction, what will be, uh, and how will be the response, they can be calculated. So Km value simply is something which tells the concentration of substance at which the maximum half of maximum velocity is achieved is known as your Km. And lower the Km value, higher will be the affinity of that substrate to uh, towards the enzyme. And the Km value always lies between 10 to the power minus 1 to 10 to the power minus 6. That's okay, I hope, fine till now. And next comes your turnover number. So it is a number of substrate the enzyme converted per minute. And there are, according to international system of classification, 6 classes of enzymes. Okay, and first class is your oxidoreductase, and its work is to transfer electron. Then transferase, it transfers the functional group from one to another. Then hydrolase, which breaks the bond by means of hydrolysis. Then lyase, it also breaking the bonds, but it is not using hydrolysis as the mean to break the bond. Then comes your isomerase, and this isomerase have uh, a process of catalysis where there is a special arrangement of substrates. And next comes your ligase. So ligase is simply for there to join or uh, combine the bonds or form the bonds. And for this reaction, they derive energy from ATP. So now, what exactly how the enzyme works, right? So first of all, what it does, the enzyme come to this, or the substrate come to the enzyme, binds with the active site, forming the enzyme substrate complex. Then it led to the formation of enzyme product complex, and finally the product is released, and now the enzyme is free of its stress and available for other substrate, right? And this is the difference key how enzyme use can lower the energy requirement for the production of the desired product. Hence enzymes are much more required for a organism. And activation energy uh, is something which is required to make the substance react. Fine. And the mechanism can be discussed in two theories. First is the uh, Induce Fit Theory, And induced fit theory can be discussed here. Ki the enzyme is having uh, a shape, but the substrate may be having a different one. For example, like here, the enzyme's initial structure and the substrate are not matching together, and hence what the enzyme does, the enzyme changes its configuration and allow the substrate to bind, and then there is formation of product. But there is also another hypothesis, lock and key. So here only one key can open the lock. So similar manner, key if the substrate having the proper step, then it will fit in, otherwise not. That means देखो, जो भी कर लो, करना है कर लो, लेकिन अगर तुम्हें मेरे टर्म्स एंड कंडीशन पे फिट हो तब भी तुम मेरे साथ रह सकते हो. दिस टाइप ऑफ रिलेशनशिप इज फाउंड इन लॉक एंड की कंडीशन. फाइन, क्लियर टिल नाउ? चलो मूव ऑन. So next is ki how the enzyme action can be inhibited. So they can be discussed under two headings, like first is the reversible, second is irreversible. Reversible means the action can be it's not permanent, it can be reversed. Okay, and this is discussed under three heading. One is competitive, non-competitive and uncompetitive. And irreversible means ki it is a permanent type of reaction. Okay, and for competitive, what happens ki here the inhibitor and your substrate both are having similar type of configuration, and both can fit in the active site. But fix, there is a fix here कि only either the inhibitor can bind or the substrate can bind. So they are acting like a enemy, two enemies, right? And if the substrate is binding, then there will be reaction, or the product will be formed. But if the inhibitor will bind, there will be no reaction. That means both are competing for a single enzyme. You can say love triangle. Okay. I know it sounds rubbish. Chalo, moving on. So in non-competitive, what happens? Ki here the inhibitor doesn't bind to the active site. Rather, it binds to some other site, and what it did, ki it simply bring change in the shape of the active site, like here, and then there will be no enzymatic reaction. And in uncompetitive, what happens, ki both there is this complex enzyme substrate complex, then the inhibitor is coming and binding to it. Then irreversible. So irreversible, it is permanent destruction or inactivation of the enzyme. For example, here the penicillin, which is antibiotic, it acts as inhibitor by binding to the enzyme transpeptidase, which is responsible for the formation of bacterial cell. And this, at what condition this irreversible inhibition can happen? First is temperature, of course, because it will denature your protein. Then heavy metals like mercury and lead, plus poisons. Then last is last type of inhibitor is your feedback inhibitor, that is allosteric modulator. So it simply influences the end product. Some allosteric enzymes catalyzes the pathway of a metabolic reaction, and they also act as the regulators. For example, your heterokinase, which is responsible for the change of glucose to glucose 6 phosphate, it is there to control the formation of glucose 6 phosphate as well, and it can inhibit the action of hexokinase. That means it will simply combine, and there will be the response yes or no depending on its presence on the medium and requirement by the organism. And functions, it's helping in simplifying the products, first breaking down of products to some smaller ones, so that there can be they can be utilized for growth and development, plus signal transduction, several biochemical mechanism for yielding energy as well, plus movement of ions across small membranes, and these are the functions of enzyme. Okay, that's all about enzyme, and thank you.